Hello，各位观众，大家好，我是图尔思仪器部的产品专员伊森。那欢迎大家参加本公司和贝克曼康德联合举办的线上讲座。那今天呢，我们将由贝克曼原厂技术人员 Eylot 来介绍一款相当厉害的仪器 BioLector XT。这是一款精心设计的高通量在线监测和控制培养的微生物反应器。那这台仪器的出现呢，它在制药、农业、环境科学、生物燃料等众多行业中带来一个全新的可能性。那并且会透过这次的线上讲座来强调它在未来的生物领域以及重要性以及潜力。那接下来就是在直播的 Q&A 环节呢，我们就是有提出问题的观众就可以获得贝克曼的14寸精美笔电包，我们是限量10个哦，所以完成提问的观众，所以请记得在那个会后的时候，我们的图尔斯粉丝团私讯小编留下您的联络方式。那接下来我们就欢迎 Eylot。
Okay, thank you so much, Ethan, for the introduction. So I think it's time to start the topic today. So I'm going to start it. Okay, so let's get started. Now from the screen, you can see the topic, and also we have I would say the most updated version, the module with the BioLector XT. You can see the integration with the Biomed electric handler. So I will go for a little bit detail on it. So I'm going to talk about the topic today. Unlocking the nature's toolbox, exploring the versatility of bioreactors. So we need a toolbox to carry the tools so that we can work on my project. So I can work on our job. So the toolbox, basically we have tools inside, okay? So now, beside the toolbox, I will send a message for you guys. So what human needs in order to have a happily human being. So what human needs? I will come to the basic. We need energy. So from the time when you wake up, your mobile phone is ringing, you turn on the lights, you boil water, turn on TV, take the lift to down your lobby, and then drive or public transport to your office. They all need energy. And then the second, which is food, we need to eat to sustain our life. And then lastly, I would say it is treatment, internal medical treatment, because eventually we will get sick or ill, so we need treatment to save us. So for the energy, the company know which is from the crude oil. So we finding some position or location. Most of the time we find some crude oil under the sea. So, but in one day they will all running out. So as a human, as people, we need to find other alternative, such as a nuclear plant. So it is more sustainable. So we find a way, but it is a matter of safety. But anyway, it is the other source of energy. So food, vegetable, we also need those to sustain our life. But and again, one day if we didn't manage it very well, so it is very likely to running out. As due to we have the technology, we know how to manage a farm. So we can have a culture of cow or sheep or chicken. So make sure it is effectively, we can keep the food source for everyone. Then lastly, which is the medication. As I mentioned, eventually we will sick, we will ill. But sometimes we need to get some medication such as drug, and we need to keep finding a new way to against a specific type of disease. That would be a good example for a couple years ago. We had a pandemic, so the COVID-19. So at that time, lot of people, researchers are looking for a way to fight against this virus. So, and again, we found the vaccine. So come to that point, and it seems that the resources that sustain human being is going to running out. So before that, we need to do something, make sure the resources which is always available for us. So that's why we need a toolbox and then carry some tools. So what does it for is make something resources which is sustainable and also effective. Okay, so back to all the thing that we need to do is for a healthy human being, we secure our basic life standard. So what we need for the toolbox, the toolbox is good, or we can generate other resources. We need energy, more energy, and even more energy. Food, more food, and lastly, we need drug medication as we will sick or ill. So this, I think it is the most important for everyone as a human being. So the toolbox, what is inside? So I'm going to tell you, actually, we have all the tools. They are on Earth. We just find a way to how to manage it, how to develop a technology to use it. They are already on Earth. We do not need to create one. They are already there. Right, so I'm going to show you what is the tools inside the box. First, we have those microorganisms such as bacteria, such as yeast. They are all in the nature. So they are ready for us to use. The matter is how we are going to manage it in order to get our goal. And then DNA. So this is a circular DNA. So I'm pretty sure every organism on Earth having the DNA, the genetic material. Now, this is the era of biotechnology, so we can get use of this technology. So you will hear the terms called genetic engineering. By putting a synthetic DNA into those organisms or microorganisms more precisely, we can reprogram them and then we can get what we wanted. Next, we need a brain. That means an idea, a researcher, the people. They come up with an idea and innovative. So we know how to use the tools. So now, back to the topic, we are unlocking the nature's toolbox. So we need a key. We need a key to unlock the box, and then we can take the tools out to help us. I would say the bioreactor, not only the BioLector XT, what I'm going for detail for today, they are the key for you to use the tools from the nature, and then you get what you wanted. As the topic for today, which is related to the bio process, I'm going to tell you the whole picture, the whole idea of what is the bio process or bioreactor is. So for example, the insulin, which is the medication material for treating the diabetic patient. So it is the whole picture to describe how to create the insulin. Okay, now we have the circular DNA we call plasmid or a vector, and then this is the insulin DNA from human. We stick them together, which is the process called ligation. And then the next step, we put it in to an organism. It is called transformation. We transform into it. For example, this is a bacteria. And then we test different types of microorganisms, maybe bacteria, maybe yeast, maybe even fungus, to find out which one get the most production of the insulin, the yield. Or even we can try different temperature, different pH to see, or different medium or the food for the microorganism to create the most yield. So now, finally, we find out one type of microorganism that produce the most insulin. And then we scale up into big bioreactor, and then finally, we purify it and get the insulin. So this is the whole idea for the production through the process called bioprocess. Okay, now, so it is the device that to facilitate the whole process, which is BioLector XT. I'm going to walk around this device so you have an idea how it looks like. From the top of the device, you can see the incubation chamber with a cover. You open it, you put the plate inside, and it is where the bioprocess is taking place. And we have the LED for the measurement from well to well, from column to column, and row to row. The next, you can see we also having another slide door right there. So it is a humidifier. It is going to maintain the humidity throughout the process. Otherwise, the cell culture is going to dry up. So to do it, we just simply put a 500 bottle with clean water, for example, Minikil. I'm going to show the video to show how to do it. Okay, so we use the probe to put inside the 500 ml bottle under the water level, and then screw it up, make sure it is tight enough. And then we put it back. It is going to show okay, and we close the door. Lights are clean. So it is the location for the humidifier. The next, we have the built-in monitor, which is touchscreen. You can touch on it to select the protocol that you are going to run. And also, you can do other options, such as start, pause, and stop the experiments. And also we can do some adjustment, such as shaking speed, temperature, gas concentration, and flow rates, as the device is going to give you a real-time result. So for example, after 1 or 2 hours of the experiment, you can see the curve, and then you feel like don't like the temperature or the shaking speed, you can adjust it in real time. So this is the panel for the touch screen. For example, if you want to increase the temperature, you just press it, and then it's going to increase immediately. Shaking speed, concentration of those gases, oxygen, CO2, and also the speed that you pump in the gas into it, the flow rates. You also can increase it or decrease it instantly. So these are the touch screen. So lastly, we have a location to put in the optical filter. Not closer. Totally, you can put six of them into this filter area. So it is for you to measure those parameters such as pH, dissolved oxygen, or other fluorescent signals such as green fluorescent protein. So this is the location. Now we also have the upgraded version. So it is called the valve control units. With this, you can conduct a microfluidic activity. In short, you can go for the fat batch study and also the pH control. So it is the upgraded version of the XT. And the other upgraded version, so we combine the XT with the network handler. So with this, you can do the sampling, and also you can do the induction without manually do it by ourselves. So the process is becoming more automated. Now, for those that you may not familiar with the BioLector technology, I'm going to tell you about the history. The BioLector technology originated from the M2P labs. We have more than 10 years of experience on the bioprocess, fermenter, bioreactor, microorganism. So in 2005, you can see the company founding on M2P Labs. So it is the company name for the Germany as a spin-off of RWTH Aachen University. It is a university that from the beginning of the M2P Labs. In the year of 2007, we launched the first BioLector. It is the BioLector 1. 2012, establishment of the M2P lab in United States. 2015, celebration of 10 years of success of BioLector microbioreactor. And also the same year, we launched the BioLector 2 and also the BioLector Pro. 2016, funding of the Asia office M2P Labs Limited in Hong Kong. And Hong Kong, which is our base, because we have an Asian region, so my base is Hong Kong, in charge for the APAC region. And then 2021, transition of the M2P labs in Beckman Coulter. So we have more than 10 years of experience on the bioreaction. Okay, so the M2P labs originated from the location called Bieswiller. Okay, so this is the map of Germany. I'm sure you one of the popular country, Belgium, and then another area which is called Cologne, and then somewhere in between. So that is the Bieswiller. Bieswiller is a quiet village and area, so it is good for study or start up a startup office. This is the M2P labs at that time. So the second level and the third level. Okay, so around 1 to 2 years ago, the office, including the manufacturing, moved to the new location, which is called Aachen. This is the Aachen city, so which is a beautiful and having, you can see that a lot of church, cathedral, and then also having other modern structure or building. So which is a beautiful city. And for the new office, we have larger space, more rooms. The room is bigger, so more people can join the meeting. We also have the facility for the entertainment, so people can chill out there and relax. And the most importantly, we have the new upgraded module for the XT, the integration with the liquid handler. We have the demo unit there. So now, Beckman Coulter and M2P Labs join forces, and then we have good resources. We have enough experience, we have connection, we have the technology, and that's why we are now having the device, which is called BioLector XT. Okay, now those two bioreactor, this is the XT, so it is a big scale bioreactor. It is the ultimate goal for the manufacturer or someone wants to make a product from the bio process. You can see this photo. We have a very big tank. So it is a bioreactor full of medium, full of microorganisms. This company working on insulin. So inside also having insulin. So it is going to running 24/7. And also for the mass production of vaccine, just like couple years ago, we need a lot of vaccine for the COVID-19. So back to this photo. So what is the relationship between the XT and the upscale size bioreactor? I'm going to show you. So from the beginning, when we want to create something, we need a bioprocess. That's what we are talking in this presentation. We start from the microorganism. This is E. coli, for example, and bacteria. From the very beginning, so the cell engineering to the final product, it involved a lot of steps. So this flowchart is the simplified one. Before that, nowadays, which is the era of biotechnology, so we most of the time use genetic engineering approach to modify the microorganism. Okay, so once we get the candidate, once we get the microorganism, we're going to culture it, inoculate it, and then move to the other step because we would like to know more information about the reaction in terms of the oxygen consumption, the pH, the medium, what type of food that we are going to feed on it, glucose or sucrose or glycerol, etc. I would say those steps you need the operator may need to repeat it hundreds of times or even thousands of times due to the complexity of the combination or the workflow. Or it depends how complex it is. So this is why we have the BioLector technology. The technology is go for the upstream process. Upstream process means before the production. We need to find out the best equation, the best formula to create the product. I would say it is going to replace those steps. As each time we can put the play in it, and then maximum we can perform or conduct 48 bioprocesses. Okay, now when we put the BioLector XT with the conventional technologies, such as the left-hand side, each time X bioreactor, each time, maybe each operator, it depends, maybe more, depend how fast the operator or the experience of her or him. And the right-hand side, it is a big size, big scale bioreactor. Each time is very likely only one or two. And also, if you look at it, each time you need to connect some external sensor, such as pH probe, thermometer, or other probe device to measure the dissolved oxygen. So with the XT, each time you can conduct 48 bioprocess, and all you need is the main units, the pressure box if you want to control the pH and do the fat batch, and you need the laptop for the software for you to do the protocol, to design the protocol, and also for the analysis after or during the experiments. Now, this is the plate that we put into the XT. For example, I'm going to use 1 well. So 1 well is equal to 1 bioprocess. 1 well equal to 1 flask. It can be 100 ml or 250. It depends your practice. So if I use up all the well, so totally I have 48 bioprocesses. It is equal to 4 shaker, and on top of it having 12 flasks. So when I compare, you can see you save a lot of medium. You reduce preparation time and also reduce the data collection time. So it is what we do. So now we know how it works, and then we back to the microorganism itself. I'm going to use the bacteria E. coli for an example because it is commonly used study model, and we know it very well. So I'm going to explain it with an equation. we input something into the E. coli, and then inside there are some processes, and after some time, outputs. Output in our term, which is product. And what we need is to refeed the E. coli, and then it processes, and then it gives a product. Okay, so back to a long time ago, 500 BC before Christ, humans know how to manage this technology. But at that time, we do not have any concept about bacteria, yeast, virus. We do not know what is inside. All we know is we put a jar of bottle on the table, and then we wait for days, wait for weeks, or even months, and then come back, and then we get the product. Okay, so there is one thing called chain or giant, depends on where you come from. So according to this equation, if I put the red grape as the input, and then after the bioprocess with the bacteria, it becoming the red wine. So the first red wine, which is called chain or kind, depends on where I come from. So that process, this technology has been happening for a long time ago. Nowadays, we know it is actually not bacteria, maybe some, but mostly it is caused by yeast. We're using yeast to make the red wine or white wine. So we put the grape juice and the yeast inside a woody barrel, and then we can make the red wine. And then it is a modern type of the barrel. So I think it is better to name it a fermenter because fermentation is happening inside. It is more convenient. We have the type, so you can easy to get the red wine. Okay, now, so we are now focused on those microorganisms. I'm going to share some application for that. Before that, as I mentioned, it is the era of biotechnology. We're using genetic engineering to do most of our job. As the DNA, which is a synthetic DNA, added into the microorganism or other organism, it is going to be programmed them, and then they are going to give up what we wanted. For example, drug. Okay, now, if we put the human insulin DNA into the microorganism, and then we feed it, and then we get the insulin to treat the diabetic situation. Back to 1951. 1,000 pounds of pancreas make 1 pound of insulin. So today, the genetically engineered bacteria produce animal-free insulin. Efficiency. 1951, how effective it is 0.01. So nowadays, which is 0.13, which is 10 times. But this data, which has come from a very purely scientific paper. So for most company working on the insulin, they have their secret equation. They have secret procedure to make the insulin, which should be far more higher than that, maybe 1 or 2% of it. So if I using the old-fashioned way to create the insulin, we need to have a culture of pig. That means we farm the pig. We have a pig farming. And then we need to hire some people to do the extraction, dissection to extract the pancreas and then do the foreign works. For now, we use a bioreactor. We put the medium, the food, with the genetically modified organism into it, and then easily we can have a lot of insulin, and then we just get it out from here. We just clean and tidy process. So it is how we make the insulin at that moment. So what if I put the DNA called IL1RA DNA into those microorganisms? It is going to produce the interleukin-1 receptor antagonist. It is for the treatment of the rheumatoid arthritis. If I put a DNA which is expressing the fatty acid synthase, it is going to create some oil, coconut oil or palm oil, so which is alternatives of those plant oil. What if I put another fatty acid synthase? You can create a fatty acid. There are a lot of fatty acid synthase. By modifying it or using different type of this enzyme, we can create different type of fatty acid, such as those. We have different type of fatty acid on Earth. With this, we can provide different taste or even different smell for us as a people. So you have heard of that. We making the artificial meat, which is come from plant, which is the plant base. By adding up different type of fatty acid, you can create different tastes of these artificial meats, such as cow, pork, or chicken, or even fish, whatever you name it, or even some new taste. So if I put the PETase, the PETase into it, so what it comes through? PET is stand for the PET plastic. Okay, so with that time, you're also giving food for the microorganism, and then it is going, and we also give it one more thing, the plastic bottle. So the plastic simply is a polymer. They stick together, becoming the plastic. Okay, by using this enzyme, it can break down into small pieces becoming different type of oligomers. In some area on Earth, some countries, they having a heavily pollution on the beach. So a lot of plastic, mainly plastic bag or bottle on the beach. It is a serious pollution. And also it affecting the sea creature under the sea. They are stuck and affecting and are being affected. Such as these sea turtles, they accidentally ate the plastic bag because it thought it is a jellyfish. Okay, if you go for the X-ray photo, you can see the plastic bag inside its stomach, and eventually it is going to die. So what if we put a COV2 spike protein into those microorganisms? If you remember back to a couple of years ago, we had the pandemic. So a lot of scientists working on the coronavirus, the COVID-19. The scientists find out that is a segment of the DNA that is going to encode and express a protein called COVID-2 spike protein. This protein enough to trigger human response, and we can raise antibody and fight against this virus without infected with this virus. By using this approach, we created COVID-19 vaccine to fight against this virus. Another, the VP1 capsid protein. So we making a virus-like particle. This particle without any genetic material, just an empty box, which is enough to trigger human immune response to fight against those viruses. And then we get the vaccine. Okay, now with this approach, we can go even further. What about the agricultural waste? Okay, we put the agricultural waste after the harvesting, such as the roots, the stem, the flower that we don't need. we put it as a food source for those microorganisms, and then we can get the biofuel. And also, it is to fill up our car, etc. Okay, next. If we only put the food to those microorganisms, then it's becoming the probiotic. So we can buy it from the supermarket. It is good for the human health. As you know, we have different types of microorganisms inside our guts. So they are good for our health. So, and again, we growing the microorganism itself as a product. We have other product, something similar to probiotic, which is called fecal microbiota. As I mentioned before, we have microorganism, which is called microbe, inside our guts. But the fecal microbiota, which is a community, like a society, there are thousands of different types of microorganisms inside. We have no way to identify each of them. What we know is they are a community, society, a lot of enzyme. We only can address a couple of them, maybe 20 to 30 of them, but actually there's a lot of different types of microorganisms inside. So for the people they're having uncomfort stomach or they're having an unhealthy guts, they may sometimes need a process called fecal transplantation. Okay, so from a healthy individual, get the feces from him or her, and then they put the feces into the patient that with an unhealthy guts status. It is called the fecal transplantation. Okay, so it is a process. So the feces from the donor, and then we mix and having some process, and then it can either put it in down the bottom by the endoscope, or create it in the form of pill, and then put the microorganism into the capsule, and then take it orally. By using the BioLector technology, we can speed up the process. We can grow the microorganism. We find a way how to grow it, which is more effective. And then finally, again, I think I would prefer the this way from the oral. Okay, now so those are the application sample for how to use the bioprocess and with the BioLector technology to speed up the whole process. And I would like to share more paper that using the BioLector technology to conduct those interesting projects. Okay, so this paper, we are not going to be through every single word. I'm going to highlight. Okay, so we're using the piquea to make the nipase, so becoming something like a drug, but I don't think it is. Yeah, I can say it is drug, but this TALAB is going to help with people with the chronic pancreatitis situation. The people not able to create enough nipase, so we need to take some nipase externally to balance this situation. So this is the paper, and then next, we using the E. coli to create a cholera toxin. So it is for the vaccine. Is for vaccine purpose. It is for drug delivery because it can create the radicals that can deliver the content into the human body. Okay, so look at this illustration. Okay, so it is the CTB, which is cholera toxin subunit B. It is form a radical, and then it is going to carry this protein, and then it delivers inside the epithelial cell, and then the protein is going to release inside and conduct its function. So it is something related with the vaccine. So next, we're using yeast to make the lipid, and then the lipid, which is the alternative way to create the agricultural oil, similar to the oil that are generated by plants. So we're using yeast to create a lipid. The other, which is we're using another yeast to make fatty acid. So it's the structure of the fatty acid. We can replace the oil from the plant, such as coconut oil and palm oil. They're making something which is comparable for the plant oil. We're also making food additives, such as L-glutamine, L-lysine, and carotenoid, something like a supplement for the human health. So it is for food additives. The next, for the bioprocess development, so we compare, in this paper, we compare 22 different E. coli, and then the scientists going to study them in terms of cell growth, dissolved oxygen, and pH level. So it is quite straightforward. And the next, we are using E. coli, we studied their expression. We induce it with IPTG, and then we study the induction time and the concentration. As those E. coli express some fluorescent signal. So, and also we use the E. coli to produce the virus-like particle that I mentioned earlier before. So this is a typical virus. Inside, it must be DNA, it must either DNA or the mRNA. And then with the virus-like particles, it's just an empty box without any DNA material inside. In theory, it only can trigger the immune response without suffering the individual. Okay, so another paper, the cell to peptide. So it is the process that the people investigating or studying the peptide. The peptide. So it is a flowchart. So now I'm going to do a little bit of summarize. So the XT, we're using this consumable. It is the plates. This plate, maximum, we have 48 bioreactions, provide real-time results. It works 24 hours and 7 days a week. Okay, then also I would like you to remember the number 3. So 3, for the XT, we only have 3 consumable. You can see we have the plates, first consumable, and the second one, which is, we call it sealing foil, which is stick on top of the plate. And then we have one more sealing foil on it, which is a silicone cover. And this is what we need to conduct the experiments. And they are the only 3 consumable for the XT. Okay, so I'm now play a video to see how it works.
In a first step, pipette the cultivation medium, the inoculum, and, if applicable, feed solutions into the corresponding MTP wells. It is recommended to transfer pH adjusting agents and feed solutions as the last step to minimize potential penetration of the solutions into the microchannels. Then manually apply the sterile barrier foil, which seals the wells off from the environment. Be sure to apply the foil properly and to attach overhanging edges to the sides of the MTP. In the next step, apply the silicone foil atop the sterile barrier foil with the help of the Foil Applicator XT. It is recommended to sterilize this tool prior to use by autoclaving or wiping with alcohol. Place the silicone foil inside the foil applicator with the brown protective film facing upward and press it down so that the pins push through the corresponding holes in the foil. Carefully remove the protective film. Place the construct atop the MTP and press it down firmly. In a last step, place the gassing lid on the MTP and push it down so that it sits in the correct position.
Okay, so it is how it works. Okay, so remember those three consumables, they are single use. So after you finish the experiment, you just throw them away. So you do not need to worry about the autoclave, the sterilization process. You just throw them away. So it can reduce the risk of contamination for the next experiments. I'm going to show some summarize the feature of the XT. Okay, so the temperature range, which is up to 50 degrees Celsius, and the shaker is inside. And totally, we can put 6 filters into it. And we have the software called BioLectors. So you can set up the protocol and also analyze the result during or after the experiments. We measure the biomass. We measure the pH. We measure the DO, which is in real time without disturbing the bio process. And we have the humidity device to maintain the humidity, 80% RH throughout the process. If you go for the upgrade, so what I mentioned before, the valve control units, you are going to add up 2 more features, which is pH control and the fed batch mode. Okay, now, so beside that, we have the new module which launched 1 year ago. With this, you can do another study. You need the first device and the second device. Look closer for this second device. It having the LED lights, so it is the emission a wavelength between 365 and 850. With this module, you can study the algae and cyanobacteria. Okay, so I'm going a little bit detail about this module. So you can study the microalgae and also the cyanobacteria. Due to the design, you can see the LED light source down below. It is called the light array module. It is for you to study the algae and the cyanobacteria because they need lights. Okay, so we come to the basic. We have a sun, and then if we talk about the photosynthesis, what we need the CO2, the carbon dioxide gas from the air and the water. And then after the process called photosynthesis, and then we have the glucose as a food and also the oxygen as the byproduct. And that's how this is the design. So that's why we have LED done under the incubator. Okay, so now is the new module for study the microalgae and cyanobacteria, and it is the module we have so far. Okay, now so beside those modules, we have some other module for the user, because when we working on microorganism, we will encounter aerobic or anaerobic study, whether the absent or the presence of the oxygen gas. Okay, so we have different module. The first one, which is default module, we're simply using the air from the atmospheric air, so from outside from the lab. And we can use nitrogen gas to flush inside the system, and then we develop the anaerobic environment for an aerobic study. CO2 up, that means we can increase the concentration of the CO2 gas up to 12%. And the oxygen down, we can decrease the concentration of oxygen from 1% to 21%. So those modules can help the researcher or the operator to study the commonly microorganism with different demand of oxygen level, such as anaerobic, microalcoholic, etc., etc. Also, we have a O2 up module, which can increase the oxygen like 100%. When you go extreme for a specific study, you can go up the oxygen level to 100%. So those are the modules that for the user to choose, and depends on their study. Okay, and another summary. So the BioLector XT, which is a screener, help to screen which microorganism is going to do the best work, and the combinator, as we have 48 wells for each run. So you can combine different type of medium, different type of pH, or different microorganism strength to see which one is going to do the best work. And optimizer, as we put different combination in one shot, so it is easy for me to optimize the process. We facilitate the whole process. And the data generator, it is going to create the result in real time and analyze it. We can study the result in the form of curve after or during the experiments. So now, this is the interface of the BioLector. This software is called BioLector. I have selected 32 wells, so that's why it shows too many curves. Okay, so I'm going to down to one well, the well D5. You can see the beautiful typical curve for those data. So for the dissolved oxygen, as it is an aerobic study, so the oxygen level is close to 0. And the pH, as we did not manage the pH, so the pH is going to maintain around 5.5 throughout the time. And you can see the biomass, we can see the exponential phase, and then stationary phase, and then that phase. So it is how we study the bio process. Okay, now the XT can facilitate the process. We can skip the long routine procedure, and we can obtain the data faster. We can minimize the hands-on time, and we can avoid manual data entry and generate graphs automatically after or during the experiments. So those are the advantages for this technology. In short, it speeds up the whole process. There's also other interesting paper from the Nature subsidiary. For example, there is a paper working on medium chain fatty acid, and there is some paper working on the biofilm by using the microorganism. Okay, now, so we have time, so allow me to introduce myself. My name is Eylot. So this is the banner that I use for the lab module that we study the microalgae and cyanobacteria. So this is my name, Eylot, and I focus on microbiology. You can see the agar plate. They on it said it is more than one microorganism. And it is how the bacteria look like under the microscope. I also have experience on the clinical lab, so it is also important for the microbiology, at least you know the hygiene technique And I also focus on biotechnology, as I mentioned many times. So now it is the era of biotech. We're using the genetic engineering to conduct most of the project. Okay, and also this picture is very classic DNA gel. So each of the pink band here, which is rich in DNA, the PCR product. Okay, so at Beckman, I'm doing presentation, so show the potential customer or other audience how the BioLector technology can assist them to complete their task. I'm also doing workshop, so people can have a chance to look at the device, they can operate it and touch it. And we also do the demonstration. And I also work with different organizations. For example, that picture I took last year with the Bioreactors. So at that time, I also would like to introduce two more fellows to you, as we work very closely. The first one, which is my direct supervisor, Joseph Leung. It is a regional product manager in charge of Asian Pacific region, in charge of particle counting and characterization, also the product BioLector. Second, the Johnson Lee, which is business development manager, also in charge of APAC region and focus on bioreactor. And again, my name is Eylot. I'm a field application specialist and also work on APAC region for bioreactor. Three of us, we work very closely together. So I hope we can have a chance to communicate with you guys very soon. It comes to the end of my presentation. Okay, so I'm going to first show you the QR code. The QR code can direct you to the link to the Bioreactors, or you can type in the Google Bioreactors and BioLector XT, and then you can contact with them. And also you can find more information of them. Okay, so see if you have a chance to scan the QR code. Otherwise, it is easy to find the name. Okay, lastly, I'm going back to the Bioreactors. I need to show you these QR codes as we have a game. So I'm going to give back the stage. We move to next session. Okay, so Ethan, thanks for the time. So it is now to next session. So please.
Okay, Alan, thank you your presentation. OK， 那在各位在正在打问题的同时，我们这边就是说，荧幕上面有一个我们 QR code 的问卷的连接。那如果说对这台仪器有兴趣的，想要来做 demo， 那就欢迎各位来扫这个 QR code。那同时或是点选下方的天文连接填写也可以。那之后我们就会安排专业的人员来与你联系，来谈论后续的实验的动作等等。那现在的话，我来看一下有哪些问题。有第一个问题是how to enable the anaerobic culture using Beckman's bioreactor? Okay, Ira.
Yes. As I doing the presentation, I have slide is showing we have different modules. So an aerobic module, which is one of it. Okay, so we connect the device. So actually there is a hole behind the rear of the XT. So just like other different, just like other bioreactor, in order to do the anaerobic, so we need to pump in the nitrogen gas, and most of the time we pump in the very pure nitrogen gas, maybe up to 99.9%, and then by keep flushing the nitrogen gas into the system. Okay, so actually on the top of the plane, we have something like a coffer, which is called a gassing need. So only one way in and one way out. So by keep bumping the nitrogen gas into it, so it can maintain an anaerobic environment. That means there is no oxygen. So it is how we managed to maintain the anaerobic development. So by using this device, you can conduct the anaerobic study.
OK， thank you。那第二个问题，second question，in one of your slide, VOWST was mentioned. I'd like to know if the production of VOWST using your bioreactor.
Okay, so before that, okay, I'm not sure if I can hang on, I have some type on. Okay, so make sure I didn't mention any other brain's name. Okay, so back to the question. Okay, so we have the customer working on it, so I cannot disclose who they are. So people are trying to using this by our XT. Okay, so I think this question is kind of referred to the last question, as it is also an aerobic study. So they trying to isolate those microbiota and then find a way, as I mentioned, the equation to increase the size, the biomass of those community of those microbes. And finally, they can becoming a drug or other form of medication for the people to use. Yes, we have one potential customer working on it, and we have conducted a couple of the experiment on that. And of course, the customer keep everything in they're not allowed to disclose the result or even how it's going, but they did use the BioLector technology to work on those projects.
OK， Eylot，Thank you。那接下来给大家看一下，第三个问题是，how to use this machine clinical microbiology.
Okay, so the term the clinical microbiology. Okay, so we need to define the term clinical. Okay, so our device is work for life science, so everything which is upstream. It is a screener to screen which strain, which microorganism is specific for some production, such as insulin, such as some oil, such as a peptide vaccine. So clinical is more like a diagnostic. So maybe I heard of some customer asking the similar question before. They may want to use the BioLector XT to do some diagnostic, because the principle, which is they need to grow different type of microorganisms. Maybe they need to put some antibiotic, put some drug to see how it works. Okay, but our device is mainly for upstream, which is for screening, optimizing, and see which equation or which strain is doing the best in terms of production. But of course, we are not limited our product to only one life science. But if I think clinical microbiology is another good topic. Okay, so we can. I think if you have time and interest in, we need to arrange another meeting, and then we can go for a detail to see if our BioLector XT can assist you with the clinical microbiology. So please contact with Ethan about to arrange another meeting. Then we can go for detail to discuss about it. Thank you for the question.
OK， 感谢阿拉详尽解说。如果说想提问的，也可以用打中文字也可以啊，我们都会帮你把问题提上来的。
可以啊。
OK， 好，那接下来的问题是 OK。What is the massive filling button of the well?
Okay, so the maximum volume of the well, so which is 2.4 mil. So actually we have a lot of different type of plays, but in general we have two main type of plays. So one which is the round well plays, and other which is flower because it's a flower in shape. For the flower, due to the shape, the maximum which is 1.9 mil. But with the round well shape, I have a demo here. So it is the round well play. So as it is circular in shape, so you can get use of the shape, the maximum which is 2.4 mil. So it depends on your study. So in short, the maximum volume which is 2.4 mil, and in terms of the volume. So as I mentioned before, we have two main type of plate: the flower shape and also the round well. The flower, as due to the shape, it can have more dynamic during the shape during the plate is shaking. So more oxygen can dissolve into it. So if your study need more OTR, more oxygen transfer rate, go for the flower plate. But in general, oxygen concentration is not a concern. Then you can stick with this plate, the round well. So as this one. Okay, so back to the question. Maximum 2.4 mil. Thank you.
OK， 那再来接下的问题。Do I need to calibrate the plate manually?
Okay, so the well we have two tiny dot which is called opto. If we talk about the dissolved oxygen and the pH, which are pre-calibrated. Before it's sent out to the customer or inside the manufacturer, it is already calibrated. And also the customer can download the calibration data. And then as this data are important, you need to update it into the software and before the experiment. And you also can download the information from the website. So we have a document for you to get the information how they conduct the calibration for DO, dissolved oxygen, and the pH. So we do not need to calibrate the plate in terms of DO and pH because it is pre-calibrated. Thanks for the question.
OK, thank you, Ella. 那还有一个，is it can change the medium when it is running the program?
Okay, so once the experiment is started, so you are not going to disturb it. Okay, so but that question is interesting. Okay, so we have the play for the 48 well for the batch study. Okay, so we also have the 32 well for the fat batch study. Okay, so if in terms of change the medium, so we can use the microfluidic, the pressure box. We can bump in the medium into the well during the experiment. But of course, the well already having the medium, and then you're going to add extra medium into it. For example, I have the well having glucose. Okay, so I'm going to shaking. After 2 hours, I would like to bump in a glycerol into it, and then after 2 hours, I'm going to bump the medium from the reservoir to the well. So finally, the well is going to have the glucose and also have the glycerol, okay? So it is kind of change of medium, but not 100% change of the medium. If you talk about change the medium, I suppose you only can add something into it. If you want to completely change all the medium, you need to get out the medium with the culture. You centrifuge it, and then you extract the supernatant, and then you get the different medium into it. But I think the whole idea is not going to disturb the bio process. I guess the meaning should be adding other medium into it. And also we have the other way to do it. As from the second slide, we have the integration of the XT with the network handler. With this, we can add the medium, or we can get rid of the medium during the time. Okay, so at that point, it depends on the operator how to set the program. For example, I have the medium in the well in the XT, which is shaking, which is, for example, I have 1 ml, and then maybe after 2 hours, I'm going to remove half ml of medium. And then I'll remove it. I've set the program with the network handler. I remove half of it. And then after some time, maybe after another 2 hours, or immediately, I'm going to add another medium into it. Okay, so it is the other way to approach if you want to change the medium. But we also need to notice that every time we need to stick with the minimum of or the maximum of the volume. But it is workable. But due to the complexity, we need to find out the way, or again, you can contact with the Toolbox to see what you really want to deal with, and see if our technology can assist your project. So I would say it is yes, but it is involve a lot of technical procedure. So it is another topic. So we need some more time to see how exactly the idea or the procedure of the process. So thanks for the question.
Okay, Eylot, thank you. Okay, 再来下一个问题。Is there any paper use this machine to perform cloning selection? And what's the principle and the pros and cons?
Okay, so paper for performing cloning selection. Okay, so cloning selection. Okay, so if doing the cloning selection, I think it's referring to different strain, as this question is referring to the main one of the main purpose of the device, because it is scanner. It's going to scan which microorganism, which strain doing the best. So if I understand this question right, so we have paper that people using the 48 well or 32 well, each time they apply different microorganism into it, different strain into it, and then after that we can see the result in terms of biomass or pH, and then according to those results, we can select which strain or which microorganism doing the best. Depends on what they are going to study. I don't think we have exactly the paper for the cloning selection, but we have the paper that provide the principle, like how the device facilitate the process, as it is quite straightforward. We have 48 well, so in principle, you can put 48 different cloning or strain into it, and then after the experiment, you can get some curves and see which parameter you are looking at. If biomass, then you simply find the culture having the higher biomass, then this is your candidate. Thank you.
Okay, Eylot, thank you. 再下一个问题。Is it necessary to use the carbon dioxide upgradation model for photographic culture?
Okay, so this question refers to the LAMP module, the light array module that I mentioned. There is a bunch of LED down the incubator. So it is for the photographic study or for the photosynthesis. Okay. So to answer this question, we need to know the concentration, the percentage of the CO2 in the air that now we are breathing in, which is around 0.04, if I'm right. Okay, so our CO2 upregulation module can up to 12%. Okay, so before that, I thought 12% is pretty much pretty much enough, which is quite high, because normally the air that we're breathing in, it only contains 0.04, which is very low. Okay, but I get to know some customer or some people in this field, they need the CO2 even more high. Okay, so it is not necessary to buy to get the extra CO2 upregulation module for the photosynthesis reaction, but I think it is good. As you can increase the CO2 because you can increase the whole process, and also I'm not quite sure if people working on it, but if we're talking about plants, we're talking about tree. Okay, so there is something called a compensation points, which is study which point there is compensation between the respiration and the photosynthesis. That means the CO2 that from the respiration, which compensate the CO2 that needed for the photosynthesis. So it is the compensation point study. But normally people work on this on plant, but I'm not too sure if people also would like to work on it on algae, on cyanobacteria. If it is the case, then why not? You need to go for the CO2 upregulation. But whether you in conclusion, whether you need it or not, so it is good to try to increase the CO2, to increase it while you're studying the photosynthesis, because CO2 is one of the key factor for the whole process. I mean, the whole photosynthesis process. So it is not necessary, but I think it is good to have. Then you will have extra data or extra information for your study. Thanks for the question.
OK， 好了，感谢你的详尽解答。那我再看下一个问题。What is the pH measurement range?
Okay, so the pH measurement range, which is from so we have two types. We have another two types of play. So which is we call a general or normal play, which is pH from 5 to 7. Okay, so it is pH 5 to 7. It is a regular play. And then we have a low pH plate, which is for pH 4 to pH 6. Okay, so at that moment, the range which is from pH 4 to pH 7. Okay, so in regular plate, which is go for pH 5 to 7, you can work on the yeast or E. coli. For the low pH plane, which is 4 to 6, you can work for lactobacillus, the lactic bacteria. So we have two types of play, 4 to 6 and 5 to 7. Thank you.
OK， 感谢艾拉。那还有其他任何问题吗？或是如果说你不好意思发问的话，你也可以透过下方的那个连结来填写，那可以把你的问题写在里面，到时候我们会再来私讯你这样子。
Okay, I'm sorry again.
No. OK， 还有 any question？
Yeah, any question?
那如果没有任何问题的话，那就是欢迎大家来扫我们的 QR code， 来就是填个几个简单的问题。那如果说评估后续的话，实验的可行性，那我们就是会来到府这边来做 demo， 来做这一台 Bioreactor 的 demo。那我们今天的线上讲座就到这里，那我们就感谢贝克曼康特艾拉的精彩的演说，谢谢大家。OK， 拜拜。
Okay, see you guys.